If you are type O, you carry a genetic mutation that has saved millions of lives and ended millions more.
Type A adds one molecular marker to the red blood cell surface. Type B adds a different one. AB adds both. Type O adds nothing. For decades, that absence has been sold as an advantage. The universal donor, the clean slate, the evolutionary survivor. But a wave of recent DNA research, including ancient genome analysis of 46,000-year-old human remains, is building a case that what type O blood is without matters far more than anyone told you. And the findings aren't reassuring. So if type O wasn't even the original blood type scientists assumed, what else did they get wrong? The name they never explained. The name type O is not an abbreviation, it's a confession. The letter comes from the German word un, meaning without. It was named that because this blood group's red blood cells lack the properties of A and B entirely. And that naming tells you more about what's happening inside your body than any donor card ever will. Karl Landsteiner first reported the ABO blood groups in 1901, working at the University of Vienna. He initially labeled the antigen-lacking group as C to denote its lack of reactivity with anti-A and anti-B sera. In 1930, he received the Nobel Prize in Medicine for this discovery, and by then, the letter had already been changed. C became O, own, because that single word captured exactly what makes this blood type structurally different from every other one. It carries no intact A or B surface antigens at all. Here's what that means in plain terms. Your red blood cells are covered in molecular flags, tiny sugar structures that sit on the cell membrane and signal your immune system. Type A cells fly one kind of flag. Type B cells fly another. Type AB flies both. Type O flies neither. The surface is essentially stripped bare, carrying only the base-level H antigen that every blood type starts with before the A or B modifications get added. In type O blood, those modifications never happen. That matters because the H antigen is not some decorative leftover. It's the foundation layer. A and B build on top of it, like different symbols added to the same base template. Type O stops at the template, and that unfinished surface state follows the blood everywhere. When was the last time you thought about what your blood type actually is, not what it means for a transfusion, but what's physically on the surface of your cells right now? The reason those modifications never happen traces back to a single point in the genome. Type O blood is defined at the DNA level by a deletion in the ABO gene, a tiny gap in the genetic code that prevents the enzyme responsible for building A or B antigens from functioning. The blood type isn't created by something being added, it's created by something missing. Blood type O, the most common blood type with a prevalence of 45 to 50%, has 25 to 35% lower levels of von Willebrand factor, a clotting protein you've almost certainly never heard of, and that prevalence means roughly 38% of the US population carries this deletion. So type O blood is literally defined by an absence. No A marker, no B marker, just a stripped-down red blood cell carrying a single deletion in its DNA. For most of your life, you've probably been told that makes it the universal donor, the safest option in a crisis. But what is that missing genetic link, and how does it disrupt the rest of your circulatory system? The protein your blood can't hold on to. That missing genetic link directly targeted a vital protein that your blood simply struggled to retain. The absence went far deeper than the outer surface of the cell itself. Specifically, researchers established that individuals with blood type O had roughly 25 to 35% lower plasma levels of a critical protein called von Willebrand factor. In everyday language, this factor acts like a specialized biological double-sided tape that grabs onto platelets and seals up tears in your blood vessels. A multi-center retrospective study published in the journal Diagnostics in 2020 confirmed this massive deficit, showing that the 25 to 30% drop was highly consistent across large populations. Here's what felt strange about this reality. Doctors knew for decades that type O blood maintained significantly lower levels of this essential protein. So why was it almost never mentioned when you asked about your blood type during a routine checkup? The clinical importance became clear when analyzing the two critical coagulation roles this protein performed. According to a paper led by Dr. Christopher C. Silliman, a senior independent investigator at the Vitalant Research Institute in Denver, Colorado, published in Frontiers in Surgery in December 2022, the protein served as the primary carrier for factor VIII, which is one of the main clotting agents in human plasma, while simultaneously initiating platelet adhesion to damaged vessel walls. Without enough of it, the baseline mechanics of healing changed, and that changes the way you should picture an injury. Clotting is not a one-switch turning on, it's a relay. Platelets rush in first, VWF helps them grab and hold, and factor VIII keeps the larger cascade moving. If one link starts weaker, the whole chain becomes easier to break. The molecular reason for this chronic deficit revealed a fascinating genetic inversion. A study archived by the National Institutes of Health in 2019 showed that non-O blood types did not actually produce more of this clotting protein than type O individuals. Instead, their VWF molecules carried complex side-chain oligosaccharides, which are essentially specialized sugar chains belonging to the A and B blood group antigens. Those sugar markers acted like armor, reducing the rate at which the body cleared the protein from the bloodstream. Because type O blood completely lacked those protective antigens on its molecules, the body cleared the protein at an accelerated pace. They lost the vital resource faster than anyone else. A 25 to 35% deficit in a vital clotting protein sounded like a sterile lab number, something clinical and distant from daily life. But what research published in peer-reviewed journals started uncovering was that this measurement did not stay theoretical when the human body encountered real stress. In fact, under the right conditions, or the wrong ones, that subtle deficit did not just make clotting slower. It multiplied the physical danger into territory that no standard blood type chart in a doctor's office ever displayed. Now, what happened when this microscopic shortage faced a sudden, life-threatening emergency? What happens when the body goes into crisis? When a sudden life-threatening emergency struck, that microscopic shortage triggered a rapid cascade of systemic failures. The theoretical deficit became an active danger the moment a patient experienced severe trauma. Under extreme physiological stress, the human coagulation system ran directly into its absolute limits. For type O patients, those critical limits arrived much faster because their baseline was already severely depleted. When you run out of that biological tape, your body can slip into trauma-induced coagulopathy, which is basically a catastrophic cascade where your entire clotting system completely collapses under severe stress. Writing in Frontiers in Surgery in 2022, Dr. Silliman hypothesized that this total failure is disproportionately triggered in type O patients. And we aren't talking about a small, isolated sample size here. A massive systematic review published in Curious pooled data from 92,661 patients to see exactly how deep this survival gap goes. They found that type O trauma patients face an incredibly high risk of hyperfibrinolysis, which in plain terms means your body's clot-dissolving mechanism goes into total overdrive, actively tearing down life-saving clots faster than your veins can put them together. The data revealed an adjusted odds ratio of 1.94 for this condition, meaning the danger almost doubled. Even worse, the same review uncovered an adjusted odds ratio of 3.02 for massive transfusion requirements, proving that type O blood struggled intensely to control their own bleeding. In other words, this isn't just bleeding a little longer. Hyperfibrinolysis means the body starts undoing its own emergency repairs in real time. Massive transfusion means doctors are no longer assisting the system. They're replacing what the system failed to do by itself. If you've ever needed emergency surgery and you're type O positive, did anyone in that room factor your blood type's clotting profile into their risk calculation? The latest population scale data suggested that they absolutely should have. Torsten Dahlén, a PhD candidate in the Department of Medicine at the Karolinska Institutet, led a massive investigation that proved these findings were not isolated flukes. His team scanned extensive Swedish health registries covering more than 5 million people to track links between ABO blood types and over 1,000 distinct conditions. They ultimately identified 49 diseases tied directly to blood groups, explicitly confirming that individuals with type O blood were far more likely to experience a severe bleeding disorder than any other group on Earth. Trauma, major surgery, and catastrophic injuries always felt like distant edge cases that only happened to other people. It was easy to look at those massive registry numbers and assume your daily life remained entirely insulated from the hidden risks of your DNA. But there was another health crisis that was not an edge case, one that impacted millions of individuals globally and proved completely predictable. In this widespread arena, researchers discovered that being type O positive did not just marginally raise your daily risks, it multiplied them by a factor so striking that it completely redefined the safety of the world's most common blood type. But what did modern genetic testing just reveal about the everyday dangers hiding inside your veins? The number they buried in the data. Modern genetic tests pulled back the curtain on a massive spike in bleeding risks that happens when specific biological conditions align in the body. It isn't a minor increase. 11. That single integer represents the adjusted odds ratio for experiencing a postpartum hemorrhage, which is severe, life-threatening bleeding after childbirth, when a type O patient develops moderate thrombocytopenia. Thrombocytopenia, which is just the medical term for a significant drop in your blood's platelet count during pregnancy, affects thousands of expectant mothers every year. When that happens, a dangerous double deficit is created. Remember how type O blood already has 25 to 35% lower levels of von Willebrand factor? If your platelet levels drop at the same time, the two primary pathways your body uses to stop a bleed fail simultaneously. It is like trying to patch a leaky pipe when you are completely out of tape and low on glue. This staggering number comes from a systematic review of 6 independent observational studies covering 92,661 participants, published in Curious. If your platelet count drops during pregnancy, which can happen without any obvious symptoms, and you're type O positive, does your OB know to watch for this specific combination? That's not a rhetorical question, but there is a crucial piece of context here that keeps this data from being pure panic. In normal, unselected obstetric deliveries, where platelet counts remain perfectly healthy, type O blood shows absolutely no statistically significant increase in bleeding risk. The danger is entirely context-dependent. It only wakes up when the second deficit triggers it. That distinction matters because it makes the signal cleaner. If type O raised bleeding risk in every delivery, you could argue the data was too broad to mean much. But this spike appears in a specific, testable scenario. The risk has edges. Medicine can see it coming. According to data published in the American Society of Hematology's journal Blood in November 2023, this blood group is heavily overrepresented among people dealing with chronic clotting issues. Specifically, type O individuals make up 61% of patients with very low levels of von Willebrand factor and 71% of those with low levels. They dominate the patient registries for von Willebrand disease, an inherited bleeding disorder where blood cannot clot properly, compared to making up only 30% of patients with normal protein levels. So the bleeding risk is real. It's documented in tens of thousands of patients, and it's context-dependent, which means it's also predictable. But here's what none of the studies covered so far can answer. Why does type O blood work this way? Why did evolution produce a blood type that protects against some of history's worst diseases, but is structurally weaker in clotting than every other type? To answer that, researchers had to stop looking at patients and start looking at bones. The 46,000-year-old answer. Researchers found the answer buried inside ancient skeletal remains scattered across the globe. It turns out our modern blood profiles are actually deep evolutionary compromises. In January 2025, an incredible breakthrough emerged when Stéphane Mazieres, a genetic anthropologist at Aix-Marseille University, published an exhaustive analysis of previously sequenced ancient genomes, which are the completely preserved blueprints of prehistoric DNA. The international study, reported by the American Association for the Advancement of Science in Science, tracked the DNA of 22 ancient Homo sapiens who lived up to 46,000 years ago, alongside 14 Neanderthals and a 98,000-year-old Denisovan Neanderthal hybrid. These samples came from ancient burial sites across modern-day Germany, Siberia, and China. What Mazieres and his team discovered is that blood type evolution was directly driven by the brutal disease environments ancient humans encountered after leaving Africa. Here is where the trade-off gets real. People carrying type O blood possessed a massive survival advantage in regions where virulent malaria was common. The unique structure of type O red blood cells made it incredibly difficult for the malaria parasite to bind to them and destroy them. Think about this. The malaria resistance baked into type O blood may have saved millions of lives across tens of thousands of years of human history. And that same ancient mutation is the reason type O positive people today have measurably weaker clotting. Does that feel like a fair trade? But that exact same cellular design came with a devastating catch. While it shielded ancient humans from malaria, it simultaneously made them far more vulnerable to other deadly pathogens. Research published in a 2021 study archived by the National Institutes of Health showed that type O blood is linked to significantly higher infection rates and severe outcomes from cholera, plague, tuberculosis, and even mumps. The human body simply cannot optimize for every single threat at once. That is the part people miss when they hear words like ancient or ancestral. Ancient does not mean harmless. It means old enough to be baked into the species. The same trait that helped one population survive repeated infection can become a liability when the diseases, the environment, and the medical world around it completely change. Mazieres noted that these deep genetic patterns raise profound questions about how far back these mutations go. Recovering this data from remains up to 120,000 years old proves that these defensive trade-offs were shaping hominid survival long before modern humans even dominated the planet. The lower levels of clotting protein aren't an accidental mistake in your biology. They are the ancient tax our ancestors paid to survive tropical plagues. So the bleeding risk in type O blood isn't a flaw. It isn't a defect. It's an ancient solution to a problem that no longer exists for most people on Earth, a biological inheritance from thousands of years of natural selection in malaria zones. But inheritance doesn't come with an opt-out. And when researchers looked at what this ancient genetic package still does in the modern immune system, who it protects, and against what, one more finding emerged that reframes the entire picture. The protection nobody explained properly. That final piece of the puzzle came into sharp focus when scientists started looking at how this ancient genetic package interacts with modern respiratory pandemics. It turns out the same architecture built to fight prehistoric threats still changes your odds in a modern hospital room. A definitive answer arrived in a massive November 2023 meta-analysis published in BMC Infectious Diseases. Researchers pooled data across 22 case control studies and 15 independent cohort studies to map out exactly how different blood types responded to the virus. They discovered that individuals with non-O blood groups had significantly higher odds of contracting a SARS-CoV-2 infection, which is the virus responsible for COVID-19. The numbers were stark, showing a pooled odds ratio of 2.13 in the case control data. In plain terms, if you had type A, B, or or AB blood, you are roughly twice as likely to test positive for the infection compared to someone with type O. This relative protective shield wasn't just a statistical blip. The American Society of Hematology highlighted independent findings from blood advances on June 27, 2023, confirming that type O individuals consistently demonstrated a lower susceptibility to catching the virus. If type O blood protects against COVID and malaria, but increases your risk of severe cholera, plague, and specific bleeding scenarios, what does your doctor actually know about your blood type beyond what's printed on your donor card? Now, does this mean type O positive provides a blanket, universal immunity to modern diseases? Absolutely not. This protection is highly pathogen-specific, acting as a structural side effect of ancient cellular setups rather than a general immune superiority. While you might hold a slight edge against a novel respiratory virus, you still carry the exact same baseline clotting vulnerabilities and historical pathogen risks uncovered across every single chapter of this investigation. What DNA ultimately revealed about type O positive blood is not a medical warning or a scary diagnosis. It is a complete masterclass in evolutionary design. Your blood type was simply optimized for a brutal, ancient world that no longer exists, where bleeding out from a wound was a secondary worry compared to being wiped out by a regional plague. The modern clotting risks and the ancient protections are not opposites. They are the exact same adaptation, a 46,000-year-old genetic inheritance that continues to pulse through your veins today. A single DNA deletion carried forward for 46,000 years still shapes who bleeds and who doesn't, who gets infected and who doesn't, every day in hospitals around the world. That's what the research revealed, and most of it was published in the last 3 years. If you found this worth knowing, drop a comment if you're type O positive and tell us, did anyone ever explain this to you?